I'm Maddie, and this is my biochemistry project on protein purification. In the beginning of the semester, we had a lecture on multiple techniques used to purify protein. I thought this PowerPoint would be a good resource to show how we apply what we learned to lecture to wet lab skills and practices. So these techniques will be shown in my work with Dr. Bowers on purifying fatty acid synthase, also known as FAS. This is a protein that is overexpressed in multiple cancer cells. So we isolate and purify FAS and synthesize proposed small molecules that will inhibit this expression in order to kill cancer cells. There are other reasons to purify proteins, and some are as basic as being able to identify structural and functional aspects of proteins. To first purify a protein, you need to have a protein. So we use the bacterial cell line E. coli to produce our desired protein. We did this by transforming the cells with our known plasmid. We grew our cells in liquid culture of LB, and we also used the antibiotic carbenicillin to make sure that we're getting the one isolate that we wanted. We grew our cells to OD 0.9 and then inoculated with IPTG, and IPTG is just used to overexpress a protein in the cells. These cells are then spun down. So this slide is just a little bit more background on how OD and cell growth are related. So OD is looking at how light passes through a cell culture. So in our case, our known OD for optimal cell growth is 0.9. So that's when we inoculate with IPTG, because this will mean that all the cells are in their stationary growth phase, as you can see on the picture of the left. And this will allow a bigger yield of our desired protein. The diagram on the right just shows the turbidity in OD of 0 and OD around 0.5. And as you can see, you can kind of eyeball it when it's in the shaker that your cells are ready to be inoculated with IPTG. So once you have cells that are expressing your desired protein, in our case FAS, we use BPER, which is a lysing agent. This just breaks open the cell and allows the contents to be expelled. This is then spun down, and the protein will then be in the supernate, which is then put through the nickel column, which I'll go over the mechanism in the next slide. The column is then washed with PBS and imidazole, which are just like neutral buffers. This will wash our desired protein into fractions, and these fractions are saved and then are used to run an SDS page. I'll go over SDS pages near the end of the PowerPoint, but for now, it's just looking at how pure your protein is. So a nickel column uses a solution known as nickel cephros. This is what we went over in lecture known as affinity chromatography. So our desired protein of fatty acid synthase is known to have a his tag. This is demonstrated in the picture below. So our protein is that circle in green, and in our case it will be FAS, and it has this his tag chain. And the thing that is binded to it is the nickel column. So when our fraction or the supernate is run through the solution, the only thing that should bind to it is these his tag proteins. This is because the his tags have a strong affinity, so like how tightly it's bound to the nickel column. So as you can see in the top diagram, loading is putting our supernate through, and it has a bunch of different things in there, different proteins, amino acids, and so forth. As that runs through the column, our protein with the his tag will bind to the nickel, and all the other things will be washed through. This is also known as the crude. The elution stage is when we wash with the PBS and the imidazole, which causes our protein to unbind from the nickel column. These fractions are saved and are considered to be potent in our desired protein, but not pure because it's still bound to the his tag and possibly other things. Our next step is dialysis. So we take the fractions, run through the nickel column if our SDS page looks good, and we put it in the snake skin in a heaps buffer. Dialysis is just used to separate dissolved molecules based on molecular weight, so we choose a snakeskin that we know will not let our protein through, but it'll allow other small molecules. So this is shown in the picture to the right of the molecules flowing out of the dialysis bag with our protein staying inside. The next step in our purification is taking this dialysized solution and running it through a DEA column. The next slide will go over the type of column this is, and it also uses another type of chromatography to better purify our protein. After this step, we also run another SDS page to see if we are getting any more pure. It's also important to mention at this step is that if we ever run an SDS page after a column and we notice that our protein hasn't become any clearer, we can go back at any point and try the previous step in order to purify the protein better. The DEA column uses a solution known as DEA cephorose. This is similar to the nickel cephorose, however it uses a different chromatography method known as ion exchange. The diagram on the left shows the structural component of DEA, where a chain is attached to a cellulose with a positive charge. Our solution from the previous step of the dialysis will run through this column and interact with the positively charged solution. Any protein that is not interacting with this will run through the column, and our protein will be left behind. Again, we will wash with imidazole and PBS to release the binding of our protein. These fractions will be saved, and we'll run another SDS page. The last step of purification is cleaving the His tag from the FAS protein. So we use thrombin. Thrombin is just a solution that we let sit with our solution that comes off the DEA column. This is usually used overnight and can be as little as a couple hours. Thrombin will actually cleave the His tag right from the protein and leave our protein separate in the solution. This will then go through another nickel column. However, this one has low affinity since we no longer have that His tag that will bind strongly to the nickel. So we're relying on the protein to interact with the nickel column. These will then be saved into fractions, and another SDS page will be ran. I would also like to mention that I typically will use only 1 or 2 SDS pages throughout this process, just so I can have a better view on one gel. I will go all over this in the next slide, but when you use multiple lanes, you can identify how pure your protein is, and you can pair them easily when they're all on one gel. So once you have all your fractions that you want to look at your protein, you will use a thing called laminate buffer. You will mix this in with your fractions, and you will then boil them. This denatures the protein so that when you run your SDS page, it affects the size, but it doesn't rely on the shape of the protein. So you load your sample in the wells, as you can see on the diagram on the left, and you usually have a buffer in there. This is known as running buffer. So you'll load your samples, and you use a positively charged anion at the bottom and a negatively charged cathode at the top. Your protein will run from cathode to anode and will run from molecular weight. So as the molecular weight is higher, it'll be closer to the top, and proteins with smaller molecular weight will be closer to the bottom. In our case, through the steps of purification, we would want to see one band, meaning we just have one protein in our fraction. In the SDS page, we run alongside a ladder. So when you get your proteins, you can see if your ladder and your purified protein is in the same spot, and you just look at this through molecular weight. The picture on the right shows what I use in my lab. So you use a glass plate that pinches the SDS page, and we usually just use a generic one that's made from a company. I think it's like Thermoscience. And there's multiple types of SDS pages, depending on like concentrations of the solution that it's made out of. Some are like half and half. Some are just all. The one we use is just one type of SDS page. So you will take this out of the package and you'll pinch it with that glass plate and put it between the green clasps. You will then fill the bottom up to the green like rubber line that runs horizontally with buffer, and then you'll also run buffer in the middle where the SDS page is, and you'll load your samples. Since you use laminate buffer, you can see when your SDS page is running, your fractions moving down the gel. This usually takes about 30 minutes to 45 minutes, depending on the voltage that you run it at. We usually use 80 voltage. So once that runs, you'll take your gel out of the package and you'll break it out of the plastic and put it in a little container. We'll use Coombs stain, which is a blue dye. You let that sit on your gel for about an hour, and we put it on a rocker so it's evenly distributed across the gel. After that, you will dump it out and recycle, and you'll use Coombs stain destaining, and you'll put that in with your solution, and this will lift the blue dye from the areas of the gel that don't have protein. So on the left is an example of an SDS page that we ran in the lab. So the ladder is this faint one right here. This would be our crude, and this would also be our crude. It's nice to have the crude on both ends compared to the ladder and compared to your samples. And as you can see, this lane and some of this lane and maybe a little of this lane has contamination of other proteins, and these are pretty pure. It's just one line. And we know that this is our FAS protein because we compare it to the gel of the molecular weight. After following our purification steps, we undergo a lot of forward directions. As I mentioned, we use small drugs to inhibit this protein, but before we can do that, we need to crystallize the protein and look for the pockets where the molecules will bind to. But for now, this PowerPoint was just to demonstrate what we learned in the classroom of how to use chromatography methods as well as dialysis and SDS pages in real-life applications. This is just to characterize functional, structural, and interactions of protein of interest. And this also just shows that purification of proteins isn't just one type of chromatography or just dialysis. It's multiple steps of purification to obtain a pure sample of your protein. In our case, at any point, we can rerun our fractions through any of these columns if we see our protein isn't pure by just checking the SDS page now and then. But overall, this project is just to show that protein purification is a multi-step process and can be done right here at Keene State College. Thank you for listening to my PowerPoint, and I hope you found this useful and maybe makes you want to join a lab.